Hello and welcome to the Wild Side. This is your host, Kight, and that was Sharon Van Etten with Afraid of Nothing. And I hope you liked that song because on today's show I'm going to be playing nothing except for Sharon Van Etten. And she is an artist that I first heard on one of Jules Holland's shows. And I thought it was a bit weird, but then I kind of investigated her album and actually ended up really liking it, and I listen to it all the time. So I thought it would be nice to profile her and see if maybe some of the listeners thought that her music is as good as I do. Now, unfortunately, there are no real obvious tie-ins between her song titles and today's topic, so hopefully you'll forgive me for not doing my usual kind of connection between music and science. But the topic that I picked is hopefully one that will be interesting nonetheless, and it's something that is kind of inspired a little bit by a listener request that I had a while back. And I had someone who was interested in thinking about GM foods and thinking about kind of human impacts on the environment and on health because of these modifications that we're making to agricultural crops and to some livestock as well. And kind of a related thing, kind of in a remotely sort of related way, is pollution from drug manufacturing, because this is another area where we're doing these kind of not incredibly well investigated things to the environment and to animals around these areas. So we know that we are having kind of chemical impacts on things and impacts on a genetic level on things, but in this particular area, this whole realm of kind of pollution and discharges from industrial areas, we don't yet quite know exactly what's happening. We get the sense that there are some bad things, but we don't yet have a whole lot of studies on this that have really shown what those bad things are and what we need to do to take care of it. So I thought it was kind of an interesting thing that links in people's interest in kind of large-scale industrial things and thinking about what companies do and how we kind of play into that to some extent by purchasing the things from these companies, and yet we can also help change the policies that are associated with what these companies can do and where they can sell their goods. So it is something that, like the GMO situation, we actually can have a little bit of control over by looking at the evidence and making decisions about how we act, and then hopefully trying to put pressure on these companies to do what we feel comfortable with rather than simply what is kind of increasing their profit margin. So that's why I picked that today, because I ran across this really nice study that is a review, and review papers are ones where the author has looked at all the literature that's out there and then summarized it. So it's actually kind of already in the format that I would use when talking about these things on the radio show. So I've been able to go through this new paper and pull out the points from the author so I can summarize those today. Now, as the author starts off by pointing out, we kind of have this bit of wisdom, this ancient wisdom, that the dose of a medicine is something that makes it poisonous or medicinal. So you can have something that's quite healthy at low doses and yet quite negative at high doses. And so this is where things get kind of interesting when you think about the manufacture of pharmaceuticals. We have these things that in humans can be really beneficial. So whether we're talking about things that get rid of infections or things that treat depression or whatever the case might be, we have things that in us can be quite beneficial. And yet the moment that those things are discharged in a large volume from an industrial pipe or whatever format they might be coming out in, those can become very negative and quite harmful because of the sheer volume of them and the sheer number of other organisms that they suddenly are coming into contact with. And so we're really beginning to think differently about pharmaceuticals, because we know that these are not things that just go into us and then that's the end of the story. We know that we excrete them at the other end, and so those things go into wastewater and can impact organisms there. And we also know that companies do their own kind of discharge and excretion, and that can be really problematic as well. And so we're now kind of taking a step back and figuring out what are the impacts of these chemicals on animals and on the ecosystems in general. And when you think about kind of the quantities that an individual person would take, or even the quantities that all of the people cumulatively in a city or in a region might take, that really pales in comparison to what we would find coming out of these large factories. And that's why this is kind of an area that is quite interesting to look at. And despite this, most of the studies have actually been done focusing on what happens when we flush these unused tablets down the toilet, or whenever we're taking something and then we go to the bathroom and flush that down the toilet. What's the impact of that level of chemical once it reaches the environment? And actually very few people have stood back and looked at what's the impact of that larger level stuff that's happening in the manufacture process. And, or potentially also, I guess I should say, in if there are any things that aren't sold, if they go unused, and so you have a large quantity of stock left over, anything like that that then has to be discarded. So any situation where you've got a lot of this stuff being dumped all at once. And this really hasn't been researched in much depth, and so we really need to understand this a bit better, because we know that there are certain toxic thresholds. That's how a lot of these things work. So in particular the antibiotics and the antimicrobials, we know that those have a negative impact on certain cells or on certain organisms at certain levels. And so if we put that out into the environment, then they can actually be doing that outside of our bodies rather than inside. And so it's quite good to know, do they reach these thresholds when they're coming out of the industries? And if they do, what then are the impacts? And it's also kind of a different scenario when you're talking about industry, because within the human body, we would take in those pills or that liquid or whatever form it comes in, and we would break that chemical down into different components. It's metabolized by us and rendered into some other form. And that other form might not be quite as problematic as the original. But when it's coming out of the industry, then you might find that actually that thing is being stuck into the water or into the soil in its original form, and then it's not broken down in quite the same way. And this could make it better, or this could make it worse. And so you have to think about the differences also in processing. So cumulatively, all of this means that the location and the nature of the impacts are quite different when you're thinking about the kind of typical stuff that's studied, so the effects of these things after they've been consumed by humans, and the type of study that needs to be done, as the author says, in the future, which is thinking about the large-scale industrial impacts. Now, manufacturing pollution can have significant impacts because of the way that microorganisms share genes, especially in the case of resistance. So we know that these guys, it only takes a little bit of mutation and a little bit of selection pressure for one or two of those little cells or those little organisms to evolve resistance, so they're adapting to this environment. And then suddenly, once they have those genes, we know that it's not just passing it on to their own offspring. We know that they can also eject those genes into the environment, and then other microorganisms can suck those up and incorporate them into their own DNA. So because they have this horizontal gene transfer, as it's called, that means that actually they can spread the trait much more rapidly, and this can become a global thing very quickly, despite the fact that these are tiny little organisms, maybe just living in one little place. And so that means we do actually have to take this kind of thing very seriously, because when you've got a big toxic dump from a chemical plant, for example, then that is putting a huge amount of selection pressure. It's really going to help these things evolve quickly, and then suddenly that kind of resistance, or whatever the unfortunate trait that we don't want to see, whatever that is, that can spread very rapidly, and suddenly we've got a big problem on our hands. And so, all of this then means that there are lots of legal and management implications. So it's not just kind of a conservation thing, which it could be if these things are toxic to fish or frogs or plants or things like that, but it's also thinking about, what are the implications for health? Who do we hold accountable for these things? Can we create incentives to prevent these bad things from happening? Can we associate costs whenever things go wrong so that we can try to extract money from people to go fix it? So thinking about all the framework to deal with these situations in which you have industries putting out this waste. So the review paper that I am going to be kind of summarizing today, it's something that looks at what we know about industrial pharmaceutical emissions right now, which is to say not really that much, but there are bits and pieces that kind of start to tell the story. And then thinking about comparing what we know about drugs versus drugs from humans, versus drugs from industry. So trying to maybe use what we know from that lower level kind of human excretion to apply that and think about kind of the worst case scenario that's possible given the industrial situation that's on a larger scale. And then using all of that to think about what our next steps need to be in terms of management and also research. So let's start a little bit from the beginning, as the author does, and take a historical perspective to kind of think about what we know already and what the state of affairs kind of is. Now, one of the things that's important to start off with is thinking about how it is that we can even look at pharmaceutical residues in the environment. And one of the really important techniques is something called mass spectrometry. And this is a technique that's evolved over the past 25 years or so, and because it is so much better now than it ever used to be, suddenly we have a much better understanding, kind of in the past few years or so, of pharmaceutical discharges. So what they are, where they are, and why they're suddenly appearing in the water and in the soil in these areas. So mass spectrometry is something that identifies the amount and also the type of chemicals that are present in a sample, and they do this by measuring the mass of these ions that are in the gas phase, and then making a ratio of that to the charge that they have, whether they're positive or negative. And these are characteristics, this ratio of mass to charge, they're characteristic of certain things over and over again. So once you kind of know what you're looking for, once you've created these profiles for known entities, you can then use that information to characterize a sample and then look for the components that you've already recognized, you've got stored away in your database. So it's kind of like fingerprinting chemicals, basically. Now, the early results showed that manufacturing discharges seem to be a source of active pharmaceutical ingredients. And this is something that I'll abbreviate throughout as API. So these are just the components of the different drugs that have been put together in the industry, and then you have excess amounts of these. So maybe if you think about it like you're baking a cake, for example, you know, as you're measuring out the flour, you're measuring out the baking soda, you're going to kind of spill it here and there. You might have a bit extra flour at the end, but it's not enough for you to keep, so you kind of you just toss it in the trash. So it's that kind of thing. It's a bit of a messy process. Sometimes you have a little bit left over. And so as they're putting all these components into the main product, they end up having some excess, and that's what ends up being discharged. And sometimes also there might be chemical reactions that have these byproducts, and there might also be cleaning fluids that are associated with cleaning up all of the area after all of these things are made. And so that washes off whatever's in the cleaning fluid might be kind of problematic, and then it also washes off the residues from the original chemical reactions as well. So there are lots of sources of these things, and they're called APIs, and these can be washed out into rivers, into streams. They can make their way into the soil because all of the discharge eventually goes out in pipes, basically. And so you've got this kind of point source of this contamination. And people knew that this was the case, but it was more or less ignored until the 1990s because it may have been kind of an eyesore, but no one really thought that it had any hugely negative impacts until the 90s, when people noticed that fish were becoming feminized. So you had suddenly a lot more female fish, not many male fish, and then they weren't able to breed. And this was traced back to point sources of contamination from industries, and suddenly people realized that sewage effluents were really important, and it was really important to keep track of what was coming from where and what rivers it was flowing into so that you could figure out how to kind of stop these problems literally at the source, so kind of solving it by cleaning it up before it reached the water, or making sure that there was some extra step installed so that it would be processed a bit more and these chemicals were extracted. Now, on top of this, industries also argued that drugs wouldn't be released, if only for an economic reason. So this kind of made people a bit more likely to ignore industry as potential cause of problem. So the reason for this is that industries were saying, well, you know, our chemicals are really expensive and our drugs are really expensive to make, and so we're not accidentally or even on purpose going to just flush down excess things down the drain, because that's really expensive to replace. So of course we're not going to be throwing this out into the environment and contaminating the environment, because it's just too costly for us to do so. So in addition to not really paying attention to things because there were no real results until the 90s that suggested it was worth paying attention to, there was also this argument from the industry that of course they wouldn't do anything like that because that would be really pointless. But once we started really seeing these hugely large-scale things like the vulture declines in India and Pakistan, suddenly we were faced with the reality that actually this is a real problem. It's happening on a huge scale, and we don't really know much about the exposure routes. With the fish, it was kind of easy to trace that upstream. With the vulture declines in India and Pakistan, which have since been linked to diclofenac residues in cattle carcasses, so these poor vultures were eating these cattle carcasses that had been laced with this stuff by some farmers who wanted to get rid of the vultures because they thought they were a problem species. And also, there were farmers that were using it for good reasons as well, and then it was just in large enough quantities that it was harmful to the vultures. You know, kind of cumulatively, all this activity was leading to these huge declines with really, really negative implications for the ecosystem and also for human health as well. And so this is when people started to actually take a closer look at this contamination process and thinking about what is going on and what is the impact of these sorts of things. And in 2007, investigations in India revealed that there were really high levels of emissions in a place called Pantacheru, and this is near Hyderabad. And this is important because this one place is a huge hub for global production of bulk drugs. And so there were lots and lots of industries congregated there in that single location, and that meant that there could be really big impacts on the environment. And the wastewater treatment plant there was receiving effluent from some 90 manufacturing units, and that meant that the pharmaceutical concentrations there were sometimes greater than those found in the blood of patients that were even taking the medicine. So this is extremely high levels, which means that all the organisms in the area were therefore being subjected to these things as well. So one example that the author gives is ciprofloxacin. Don't try to say that one fast. And that one's a broad-spectrum antibiotic. And they were finding that levels of this were as high as 31 milligrams, or 1 million times greater than levels that were found in treated municipal sewage. It's toxic to a lot of organisms, as you might expect, given that it's an antibiotic, and the estimated total release of this chemical per day was about 44 kilograms, and that is sufficient to treat everyone in a city with 44,000 inhabitants. So clearly this is a huge amount, which is not only incredibly wasteful, but it's also incredibly problematic if you don't want microorganisms to start developing a resistance to this, and you also don't want it to contaminate and potentially harm other types of organisms as well. And the researchers found that this was polluting river sediment and surface and groundwater, the drinking water, and even the irrigated soil. So it was just going all over the place where it could impact lots of different organisms. Now, unsurprisingly, these results attracted a great deal of media attention, and that then increased the interest of researchers and also just kind of regular folks who were paying attention then to what was happening in their environment. And similar things have been found also for oxytetracycline, penicillin, ethinyl estradiol, and other such chemicals in association with factories in China. And in these places there was contaminated treated effluent, so this stuff was being treated, but it wasn't really being treated enough. So these chemicals were still escaping from the factories into the habitat, and they were going then into the surface waters that were running nearby, so it's contaminating the water in the area. And the hormones from these things were, some of these chemicals were hormones. I said the ethinyl estradiol, for example, and these levels were sufficient to disturb the reproduction of aquatic vertebrates, which again can have a really big impact on the functioning of the ecosystem. Since these kind of original studies came out, a number of studies have now found other concerning pollution patterns all around the world, not just in the East. So there's in Asia, all over the place, but also in Europe and the US, and a lot of these places have been found to have kind of uncomfortably high levels of APIs. And in Europe, in case you're interested in thinking about places closer to home, some of the studies that have been done have investigated places in Denmark and Germany, Switzerland, Norway, Spain, and Croatia. I didn't see any on the list for the UK, so that's good for us. Some of the focal drugs include things like phenazone, which is a non-steroidal anti-inflammatory, venlafaxine, which is an antidepressant, and also some sulfonamide antibiotics. So basically these are all sorts of classes of drugs. You know, we're talking about painkillers and antidepressants and antibiotics. It's a little bit of everything that we're talking about, not just one class of drug. And some of these studies have analyzed streams with mixed inputs, so places where you've got municipal sources but also industrial sources. And so it's quite hard to take the results of those studies and figure out exactly what patterns are a result of the industrial input, because you've got this, you've got this stuff coming from both of these places, and so you can't tell which is which unless it's extremely high levels. And if you've got an extremely high level, it's unlikely that municipal sources would be the cause of that, because we just don't use the antibiotics and the other drugs in levels that are high enough to create these big spikes. It's really only the industries that pour things in that amount. The only time you'd ever really see that is if you've got maybe some kind of an epidemic of something, which wasn't the case in these studies. So the researchers have begun to develop ways to use the ratios of component compounds to try to separate the two, but this is still a bit nascent. They're really trying to refine this even further to better figure out what is the source of these different things, and then how to kind of go and fix that. Now, despite these sorts of studies that are kind of, getting a preliminary idea of what's happening, we're still lacking lots of information. One of the things we want to know is how common are large emissions of APIs. At how many of the factories can we expect this to be happening? Where are the APIs being dumped into the environment? So is it typically going right into a river? Is it going into creeks? Is it happening on the soil and then filtering through the soil? So to get a better idea of kind of the flow. Also, figuring out which APIs are problematic. It might be that some of the chemicals are bad, or it might be that all of the chemicals are bad. So you need to figure out which ones are causing problems and for what reasons. And in particular, you need to kind of know from start to finish what are the different impacts of these APIs once they reach the ecosystem. What are the organisms that are impacted, in what ways are they impacted, and how can we try to mitigate that? Now, what's kind of interesting is the author does point out that there has been one study that suggested that actually the industries aren't really responsible for that much contamination at all. There was one study that happened to have been commissioned by a pharmaceutical company that said, we've tested all of our effluent and we have not found very high levels. Now, they may have found this because they were commissioned by pharmaceutical companies, and so of course they're going to find results that are favorable for themselves. But it also is true that negative findings are quite hard to publish in the scientific literature. People like things that have a negative result because it's much easier to say this didn't happen than to definitely state for sure, I found these results and therefore this is happening, this is the pattern. And so we tend not to publish things that are negative findings. And so people might have actually had negative findings from all their other previous studies, but they just weren't able to get them studied. They might just be underreported. And so it's going to be important to kind of go back and see if we can rummage up some of these studies, talk to the authors who actually did them, and then also make sure that if the findings were negative, that was because the researchers were actually having truly an absence of things in the water, and not because they were testing for the wrong things or doing some other kind of methodological thing that makes their research not quite as useful. Now, at the moment, where things stand is that API emissions aren't really that commonly regulated, and companies don't often self-report. They're not required to do so, and emissions data aren't really publicly available from the companies themselves or from the authorities, and so overall we just don't have much information, and we need to try to figure out how we can improve our monitoring and how we can then not only collect those data but then also analyze them and publish them in order to try to help make our management strategies and our legal strategies a bit more efficient. And we also need to investigate not just the transmission of these things through the water, but also potentially through solid waste and through air. And with that thought, the author concludes his kind of background contextual part of the paper, and then transitions into thinking about some of the studies that have actually been done and what we do know from the little research that there is out there. And I will go into that in just a minute, but first I'm going to give you a bit of a break with another song from Sharon Van Etten. Welcome back to The Wild Side. This is your host, Caitlin Kight, and that was Sharon Van Etten with Our Love. And on today's show, I am talking about a recent review paper that looks at the effects of pharmaceutical waste on the environment. And just before the break, I was giving a bit of background, and now I want to think about some of the few studies that have actually been done on this topic to summarize what we know. So there are several studies that have been done that have looked at how the wildlife around these industrial areas have been affected by exposure to effluents containing chemicals from drug manufacturers. Some of the studied species include planktonic bacteria, marine amphipods, fish, crustaceans, mice, water fleas, plants, frogs, rats, bacteria, and kind of general microbial communities overall. So obviously there is quite a range here. Now most of those are much smaller organisms and not the larger organisms up the food chain, which we know can be impacted. So for example, we know that mercury contamination from industrial sites has affected not just the little things, but also the larger animals because it's gone up through the food web. So that's quite possible here, but we're kind of, I think, still at the beginning stages of these sorts of studies, and so we're starting small and looking at where these chemicals are potentially even entering the food web to begin with after they're dumped by the companies. Now, some of the studies also provide information on exposure concentrations of the APIs, of these components of the products, of the ultimate pharmaceutical products, but mostly there is little direct connection that's made between the presence of APIs and the actual effect of the effluent. So we kind of have this general sense that there are some things that are affected and that there's something that's coming out of the industry, so we have this kind of nice correlation that looks convincing, but nothing that's really causative yet, and they've not really been able to collect these hard data that can really help solidify this link. Now, one example that has been made is the link between antibiotic-resistant bacteria and antibiotic-contaminated effluent and sediment. That's a pretty clear-cut, obvious one to look for. There's also been another example that I kind of referred to earlier, which is where you've got this problem with fish. I was going to say with fish sex. It's not really with fish sex, but with the hormones that are affecting the gender and also the ability of these animals to breed with each other. And this has been found both in the U.S. and also in a study that's cited here that's in France, so looking at effects downstream of a steroid factory. And again, these are situations where you kind of get a pretty good idea that there's a lot of evidence here that this is in fact a cause and effect thing, but still we could use some additional research that really has good quantification and kind of finds a threshold for the level at which suddenly these chemicals start to become a real problem. And another thing that the author notes is that even though these components of the drugs are quite powerful and can have huge impacts, there are also other compounds in the effluents that could be equally impactful. And this is kind of what I was referring to when I mentioned the fact that when there is cleaning that goes on, the cleaning fluids themselves, as well as the things that are cleaned out, can be problematic. And also you can have compounds that result from they're kind of byproducts. Whenever you mix two chemicals together, you might have a bit of a discharge that's not in the drug itself, but it's kind of a byproduct that's then gotten rid of and disposed of in these effluents. Now, one example of this is phosphoric acid, and this was something that is not an API, but it's in the effluent anyway, and it's found to have been killing fish in a Norwegian river that was receiving effluent from a pharmaceutical factory. Similarly, in China, there were some 60 factories that were closed to protect water quality, and all of them were producing something that is a steroid precursor. So we know that there are these things in the water that aren't just the APIs themselves that can be quite problematic. So how do we know what's happening in the wild, given that we don't have a whole lot of research on this? Well, a few studies have found adverse effects of components in the field. Others have done laboratory analysis of levels that have been found in the field, and then they kind of use that information to then set up actual experiments to test it in a laboratory setting. So basically you've got kind of a combination of observation that's happening out in the wild and then testing that's happening in a laboratory setting. But it's quite hard to kind of extrapolate the effects of the chemicals unless we know exactly what the recipients are that could potentially be interacting with these things. Different species might be sensitive to different levels of the chemical, and so you have to test individual organisms separately in order to gauge the impacts on wildlife. And then you have to think about the organisms that those guys go on to interact with and what role they play in the ecosystem to really figure out those broader level effects. And obviously this is quite a huge proposal then. There are all sorts of things you'd have to do. Now, one thing to keep in mind is that if you have certain keystone or umbrella species that are kind of disproportionately affected, or that themselves go on to disproportionately affect the ecosystem, then maybe you could start to pull out one or two kind of indicator species per habitat. But you'd still have to know something about that habitat and about that ecosystem to figure out which animals or organisms you would want to focus on to do that. Now, it's also important not to get distracted by thinking solely about lethality. So obviously, if you go out and see a bunch of fish floating belly up in a stream, you might say, well, that industry up there is having a really obvious negative impact. But there are lots of things that can happen that don't cause death, but are still really problematic. So if you're impacting gene expression or enzyme activities, this can have long-term implications by reducing an animal's condition, reducing its health, reducing its ability to breed, and therefore reducing the fitness of an organism or a whole population of organisms, which means that over time those animals would start to tail off, so that even if you don't see a negative effect that's really obvious right now, in a couple years or even 10 years, suddenly you might find that those animals aren't found there anymore. And so you have to think on a different scale as well, and think about how these chemicals act to figure out what the mechanisms are by which they might actually be having these kind of physiological or reproductive sorts of effects. Now, lots of studies, probably the majority of studies really, have focused on bacteria. And one thing that's kind of problematic about this is that you can't see them with the naked eye. And so you're talking about activities out there in the wild that aren't going to be really obvious. You might not just look at the environment and say, oh, yes, things are going terribly here, look, everything's gone, because you can't see the bacteria to start with. And it's quite hard then to conduct field observations and to necessarily know which areas need to be targeted because you don't really see necessarily that anything is amiss. So we need to also think about kind of sampling these larger organisms that might also be impacted, and thinking about, are some of these things absent because the waters are too toxic for them? And this is another thing which might get kind of difficult, because you might go to a place now and look around and survey it again after a big discharge and look around, and it looks kind of the same. And what that might reflect is that the stuff that was being impacted has actually already been killed off, or it's already left. And so you're not able to see a big difference because only the stuff that is hardy enough to tolerate it is now found in these areas. And with bacteria, that's certainly the case, because they do adapt so quickly that suddenly it might be the case that the things we're sampling, they all look pretty good because they've already adjusted to that kind of toxic environment. So you have to keep all these things in mind when you're actually doing the research out in the field. Now, antibiotic resistance is quite an interesting case, because normally it's considered kind of a beneficial thing, so it's a sign of resilience, it's a sign of adaptation, but that's positive from the view of the bacteria, maybe not so much from our standpoint, because it's great to know that some wildlife are able to cope with the things that we do to the environment, and yet in this case it's quite a dangerous thing. So if bacteria suddenly have antibiotic resistance, then if they infect us, it's very hard for us to treat them. And even if these are bacteria that don't infect us, that's still a little army of organisms out there that can pass on those genes to things that might affect us in the future. And so it's quite a scary thing to have these genes increasing in the habitat. So we need to get a better idea of exactly how much exposure is causing these sorts of responses that we're seeing in bacteria and other organisms, and how fast the microbes and the other species are responding once they are exposed. So what's kind of the speed with which they can adapt and begin to evolve. And it would be quite useful to examine this experimentally, not just by doing some observations, or at least to do much more complete chemical characterizations of the effluents, and then the communities around them, and then kind of the relationship between the two. This is especially true with bacteria, because you can culture these guys and grow them up in the labs as well as out in the field, so you can start to correlate what's happening in real life with what you can do experimentally. Welcome back to The Wild Side. This is your host, Caitlin Kight, and that was Sharon Van Etten with Break Me. And just before the break, you'll notice I had quite a small section there talking about the studies that have already been done on the effects of wastewater effluent from industrial areas making pharmaceuticals and what the impacts of these are on wildlife and the ecosystem. So actually not much is really known because we're only at the tip of the iceberg here in terms of performing this research. But you can take that and then kind of figure out then, okay, what don't we know, and therefore where are the biggest risks? And one of the things that the author focuses on is the fact that the risks associated with discharge from industrial areas are differing from those associated with the excretion of drugs by humans, mostly because of variations in exposure levels. So you can't necessarily just scale up, because we know that the exposure levels are hugely different, and therefore it could have big impacts on the wildlife. And this is because in sewage you've got concentrations that are quite low, because only a few of us in any given area at any given time are going to be using a particular drug, and then we break it down, we flush it down the toilet with water, and the water dilutes it, and we also take showers, we clean dishes, we wash our cars, all these other things that we do. We typically use a lot of water here in the West, and so we're incredibly diluting the sample of what actually reaches the wastewater treatment area. And then we tend to also have really good treatment practices that are removing a lot of these chemicals before it's ever released from that area into the wild where it can interact with wildlife. So, municipal waste is actually going to be not much of a problem relative to this big industrial stuff. What's kind of scary about the industrial waste is that you often will find that the majority of the world's production of a particular API is going to be found amongst only a few factories or in one single factory. And so you have this huge point source of just tons of a particular chemical, which can be obviously really problematic for the environment. And the levels found in these areas might be just a fraction of all the APIs that are handled by the facilities, so you might not see contamination from absolutely everything, but still you could find that these things are much higher than they would be found in municipal waste, and therefore potentially much more problematic. And this might be enough to have a high enough concentration to actually trigger some sort of a response in wildlife, and that's what we're really interested in knowing. Another kind of twist on this is that exposure patterns for wildlife can be potentially quite erratic, because drugs may not be produced year-round. There might be little pulses of production. There might be an association with when the tanks are cleaned periodically, and so when you use the cleaning fluids and wash out certain byproducts, suddenly you've got a big peak of exposure then as well. And so you would need to know exactly what the routine is in each of these facilities, so you can regulate and monitor in a much more known way and kind of have that schedule set up in advance, rather than just relying on being able to go any old time and sample the water and get an idea of what's happening in that area. So the next thing you really need to do is identify which of the APIs are posing the greatest environmental risks. And it's quite difficult to estimate industrial discharge concentrations without information from the companies themselves about how and when they're working with the chemicals. And clearly, this is probably not a thing they're going to be incredibly willing to just hand over. And we would need information, too, on what happens to the chemicals after they reach the environment. Are they degraded? Do they hang around? If they hang around, how long does it take for them to then be broken down in some way, if ever? If they are degraded, are the resulting products also harmful, or are they kind of suddenly just rendered inert? Where do the chemicals go after they're degraded, or if they stay in their whole format? Are they deposited locally? Are they transported elsewhere? You have to know all the dynamics of how these things move through the environment. And it's important to keep in mind that even the things that aren't typically thought of as harmful could have adverse effects if they stick around long enough to cause an adaptive response in wildlife. And so everything really should be should be considered potentially dangerous until you rule it out and make sure that it's not having these bad effects. And to date, we don't really know about any distant impacts of discharges. We've just studied stuff that's kind of near the point source of contamination. But we do know, in the example of microbial resistance, that a response only has to kind of pop up once for it to have an impact. And if it travels even just one little organism downstream, then it can have a big impact elsewhere. So we do know that we should kind of open our net a little bit and extend it a bit wider, so that we can look not just right next to factories, but elsewhere as well. And certainly the vultures in India are a good example of that as well. And kind of an interesting thing to think about as well is the fact that this is both a global and a local problem, because the the manufacturing companies might be in India or in China or wherever, but those drugs are then sent here to the West where we ingest them as well. And so, we don't have to feel guilty about this, but it's good to be informed, because certainly if we know about things and we can put pressure on our governments and on these companies from over here, then we can help make sure that the environments over there are quite healthy. And a great example of how this can actually work is what's been done with coffee growers, and thinking about that kind of, the fair trade attitude and the eco-friendly attitude. And we have asked manufacturers to make these changes, and they've done so. So we know that this is a step that we can take. And one of the things that the author suggests is that we need to have regulations for surface water levels of API. And the European Water Framework Directive, that's quite a mouthful, the EWFD, it has something called a watch list, which includes things like diclofenac, which is what affected the vultures in India, and estradiol, which is what was affecting the fish. But it only has developed these more general warnings, and it doesn't have actual numerical guidelines, and it can't really punish things either. So this is something that we can use as a tool to start off with, but it's not really a regulatory body, and it doesn't really help us actually go in and actually make a change. And so this leaves the municipal sewage treatment plants without specific goals. It leaves the industrial plants without specific goals. And so we need to have kind of a real quantitative shift here, so that we can provide some numbers that these guys can aim for, and they can look to see if they're falling within the right boundaries or not, and try to rein themselves in if they aren't. So in order for American and European countries to influence the activities of companies outside their borders, they could potentially amend something called the Good Manufacturing Practice. And this is a framework that could be including some environmental criteria that could inhibit the import of products that aren't produced in this way. And the pharmaceutical company AstraZeneca has already proposed a model that might be adaptable for this purpose, but it doesn't address some of the things. So, for example, antibiotics and resistance aren't included in this. But it's a start, and it's nice that it's coming from within, from a pharmaceutical company. So organizations that purchase and ship and invest in the pharmaceutical products, they could all be responsible, and we could be responsible by buying into them, for offering incentives and offering kind of bonuses and tools like this that would offer a positive reason for these companies to change. And we could also have some kind of negative things as well. So the media could cover the stories when the companies aren't doing what we're asking, and there might even be some fines as well if they exceed certain limits. But the nice thing about this is that it could be led by the governments, and they could help the industries do what they're doing. And since governments are, hopefully representative of us, and because we can have tools that help us interact with the government to persuade them to do what we want, then we could actually lead this movement as well. And that's part of the reason why I wanted to talk about this on the show today, because it is an example of something that, the listeners of the show have clearly indicated that they think this kind of issue is important, and it's something that you guys can then use this information to go out and maybe do something about. So hopefully if you are interested in this issue, you can follow that up and try to encourage your representatives to make sure that companies like this, not just pharmaceutical companies, but really any company, are being eco-friendly and making sure that their products aren't hurting the environment. So on that very nice conservation-minded note, I'm going to wrap up with my final song from Sharon Van Etten, which is Every Time the Sun Comes Up. Enjoy, and I'll talk to you, which is Nothing Will Change. Enjoy, and I'll talk to you next week.